Endothelial lipase is a recently discovered enzyme that belongs to the triacylglycerol lipase family. In this presentation, we will explore the structure and function of this critical enzyme. Endothelial lipase was first isolated in 1999 by the Rader lab at the University of Pennsylvania, where researchers determined its amino acid sequence, found its sequence homology with the other members of the triacylglycerol family, hepatic lipase and lipoprotein lipase, and began to characterize its substrate specificity. Researchers also started to examine the overexpression and underexpression of the enzyme and the effect that these manipulations have on lipid metabolism in mice. They showed that endothelial lipase, EL, alters blood levels of high-density lipoprotein, HDL, a circulating factor in the blood that may correlate with reduced heart disease risk in humans. The results shown in this graph illustrate the proposed function of endothelial lipase. When Thomas Kurdermos and colleagues created knockout mice for the endothelial lipase gene, the amount of apolipoprotein A1, the main protein component of HDL, increased significantly in the knockouts as compared to the wild types. This association reveals the critical importance of endothelial lipase as a negative regulator of HDL metabolism, and since then, many studies have been done to illustrate the structure, function, and therapeutic implications of this enzyme. Endothelial lipase, in its active conformation, is a head-to-tail homodimer. Immunoprecipitation experiments performed by the Rader lab revealed that monomers of endothelial lipase oligomerize into dimers. Further experiments with ultracentrifugation and radiation inactivation confirmed that endothelial lipase exists as a head-to-tail dimer. The y-axis of this graph indicates the amount of endothelial lipase activity, while the x-axis describes the amount of inactivation of the enzyme caused by radiation. The results were consistent for both wild-type and constructed endothelial lipase in that both functioned maximally in the absence of cleaving enzymes and performed worse in high concentrations of cleaving enzymes. The similarity of these results point to the accuracy of the claim that endothelial lipase is a dimer. The dimers are linked by a hinge or short chain of 8 amino acid residues, which include glycine, serine, isoleucine, glutamate, glycine, arginine, leucine, and glutamate. Aside from the quaternary structure, endothelial lipase contains a serine esterase catalytic triad of serine, aspartate, and histidine located in the N-terminus of the enzyme. Structurally, endothelial lipase is extremely similar to other members of the triacylglyceride lipase family with critical roles in lipid metabolism, hepatic lipase, and lipoprotein lipase, LPL. All three enzymes are homodimers, and it is known that LPL also exists in a head-to-tail conformation. They also each have a serine esterase catalytic triad which hydrolyzes their substrates. What distinguishes each type of lipase, in addition to their different distributions of expression throughout the body, is the C-terminus structure and the lid domain, which determines the type of lipid that the lipase will interact with. Endothelial lipase interacts with both triacylglycerides and phospholipids, and it most effectively hydrolyzes high-density lipoprotein, with this substrate specificity encoded in its N-terminal domain. High-density lipoprotein is made up of both proteins and lipids. Apolipoprotein A1 is the main protein component, and it interacts with a variety of different lipids. Phospholipids are an important part of HDL structure because they have a very polar head which allows the HDL molecules to circulate in a polar environment of the blood while carrying multiple nonpolar lipid species. Phospholipids have 2 fatty acid chains, one saturated and one unsaturated, and a phosphate group which are all bound to a glycerol group. Endothelial lipase specifically binds to one of the fatty acid components of the HDL and cleaves it, leaving a free fatty acid. It cleaves the phospholipid at the SN1 position, or the chain that is furthest away from the polar phosphate group at the head. Phospholipids are an integral part of the HDL molecule, and by cleaving the fatty acid group, endothelial lipase is breaking down and ruining the phospholipid component. The phospholipid component of HDL is crucial because it protects the apolipoprotein A1 component from being filtered out of the blood by the kidneys. When endothelial lipase breaks the phospholipid component down through cleavage, the high-density lipoprotein is now cleared out by the kidney, lowering its concentration in the body. When it was discovered, endothelial lipase offered a novel and exciting opportunity for therapeutic efforts focused on raising HDL levels to prevent risk of heart disease. Because endothelial lipase lowers the concentration of the protective HDL in the body, it is an important component of the discussion about therapeutic treatment for cardiac disease. HDL travels through the body and carries lipids away from plaques in the arteries and delivers them to the liver, where they can be converted to bile acids and excreted by the liver. This makes it a very attractive therapeutic target to mitigate the risk of coronary heart disease. There is some disagreement in the scientific community as to whether endothelial lipase activity is actually harmful or if it is necessary and natural for the functioning of the body. Currently, research on endothelial lipase is being done with an emphasis on exploring this dichotomy and is attempting to determine whether the inhibition of this enzyme in humans would be an effective preemptive treatment for cardiac disease. The implications of this question are enormous and could change the lives of millions of people around the world.